Hello everyone, thank you for watching this pitch. I'm Helen Carlson, a researcher in the Department of Pediatrics within the Cummings School of Medicine at the University of Calgary. I'd like to take a few minutes to tell you a little bit about the MOSAICS toolbox and what we are challenging our innovators to do. The first thing that I'm sure you've noticed is that MOSAICS has a great big long acronym: Mapping Sensorimotor Activation Hotspots During Cortical Stimulation. So we'll just stick with MOSAICS for short. Non-invasive brain stimulation tools such as transcranial magnetic stimulation, or TMS, allow us to interact with the brain. We can turn a small region of the brain on or off and measure the behavioral changes to determine the role of that brain region. TMS is a powerful tool to investigate the motor network. In the example shown here, we can apply TMS to various places on the scalp over primary motor cortex, measure the size of the resulting hand twitch, and then use MOSAICS to create a heat map to characterize motor cortex. Now, people are pretty similar in how their motor cortices are organized, but after brain injuries, like having a stroke at birth, they can be very different. In our lab, we use TMS to investigate motor cortex reorganization in children with perinatal stroke and cerebral palsy. We can use TMS to look at how the motor network has changed due to stroke and how the brain compensates for this injury. We've created the MOSAICS toolbox to aid in this process. MOSAICS creates these motor heat maps from TMS data and transforms them into a standard space so that we can compare maps between individuals and within different individuals over time. MOSAICS is a one-of-a-kind tool, but it's not really easy to use at the moment, and we want to add more functions as well. So we have a great opportunity here for a team of innovators to elevate MOSAICS into a fully functional toolbox, including a graphical user interface, better error checking, and other quality improvements. Once the MOSAICS toolbox is established, exciting new functions can be added, including additional imaging features such as automatic skull stripping and group-level analysis. The ultimate goal for this project is to release the MOSAICS toolbox for use by other labs across the world. Innovators on our team will have the opportunity to learn more about brain stimulation and brain neuroimaging. They will co-develop this toolbox and possibly earn a co-authorship on a publication supporting this toolbox release, should they be interested. Finally, learning to collaborate on and implement others' code is a universally useful software skill. So this is a great project for anyone interested in software development. So come chat with us during the team formation event to learn more about how you can get involved in the MOSAICS program.